Cancer preventive vaccines for Lynch syndrome carriers, how do they work? Increasing longevity of humans comes along with increasing incidence of cancer. To keep our organisms functioning at an advanced age, our cellular mechanisms become less stringent against genetic alterations. Accumulation of a certain combination of genetic alterations or mutations might lead to loss of control over cell division, clinically manifesting as a tumor. However, this process usually takes many years, which is why most tumors occur in elderly persons. However, sometimes cancer can occur at an unusually young age. The most frequent reason for this is a genetic predisposition to tumor development. This means one of the genetic alterations required for tumor development is already present in the fertilized egg cell. Thus, in individuals with germline genetic alterations, tumors develop faster. The most common inherited cancer syndrome is Lynch syndrome. Carriers of Lynch syndrome have a genetic alteration affecting the mismatch repair (MMR) genes, which are responsible for correcting small base mismatches during DNA replication. The pre-existing genetic alteration in Lynch carriers enhances the risk of acquiring further mutations. Lynch syndrome is estimated to affect up to 400,000 individuals in Germany and 3.5 million people in Europe. Carriers are at a 5 to 15 times higher risk of developing cancer, particularly frequently manifesting as bowel and uterine cancer. They require specialized prevention, such as an early colonoscopy. The unique mechanism of tumor development in Lynch syndrome that is tightly linked to a natural immune reaction against tumors allows to design a vaccine, which could prevent Lynch syndrome tumors. The human genome consists of 3 billion base pairs, and during every cell division this entire genetic material needs to be copied. As mistakes occur during every cell division, our cells are equipped with DNA repair mechanisms. The MMR system is one of them, specializing in the repair of mismatches. The repetitive DNA fragments, so-called microsatellites, are particularly prone to mismatches. Mutations occurring at microsatellites within coding regions of the genome have major consequences. As the sequence is repetitive, one additional insertion or missing deletion base leads to a shift of triplet sequences, needed for the translation into an amino acid sequence that subsequently results in a protein that is substantially different from its originally encoded variant. If the encoded protein has a function to control cell growth and division, then a mutation can make the protein unfunctional and result in tumor development. Moreover, the newly generated protein is not only unfunctional, but also completely foreign to our immune system and contains several immune-relevant antigens. This is why the immune system starts producing cells and antibodies directed against these antigens. As it is defined by evolutionary forces, which particular mutations can be favorable for tumor development, we were able to identify major gene targets commonly affected by microsatellite mutations. As it is also possible to exactly predict the proteins which result from certain mutations, we deciphered the antigen profiles of tumors arising within Lynch syndrome. With this information, it is possible to design a vaccine that could inform the immune system about these antigens prior to tumor development, and thereby possibly prevent tumor development. The efficacy of such a vaccine must be validated through large-scale clinical trials.